Hello again. So by this time, possibly you've reviewed hydrolysis and condensation reactions. Another term for condensation reaction is dehydration synthesis. That's right. Good job. Good job. So now we're going to review nucleic acids and then in general, all the macromolecules. All right. We'll cover nucleic acids in much more detail, possibly at the end of this week or next week. We'll see, but at the least, we'll be familiar with the structure. All right, so we know it's a double helix. Its general function is to store and transmit hereditary information. All right, so what is that information? You already know this. That's right, how to build a protein. And a few other things, but primarily DNA's function is the information of how to build a protein, because you are proteins. If I took, excuse the example, but one parent and put them in a blender, and the other parent put them in a blender, and I took you and put you in a blender, and we compared each other, and what you'd find is that you each had very similar proteins. You would have a blend of the proteins of your mom and dad, and that's just because you would have had the same or similar DNA. And it's the DNA that codes for the proteins. Anyway, RNA, let's just say pieces. RNA and DNA are the two primary examples of nucleic acids. There's nothing else to it. They're really, really long. Like if you blew it up to be the diameter of a hair, it would be the length of a tennis court. Is one analogy I've seen in a textbook. But the basic structure of nucleic acids are the nucleotides. These are the monomers. The single pieces of nucleic acids are nucleotides. Nucleotides are made up of three parts: a nitrogen-containing base, which is this yellow portion. It can be a double ring or a single ring. A pentose sugar, that is a 5-carbon sugar. You'll see that right here. Oxygen at the top. Carbon 1, carbon 2, carbon 3, carbon 4, and there's the carbon 5. That's the 5th carbon right there. So you got a 5-carbon pentose sugar, and then you have a phosphate group. Now this is not to scale. They've made this really large, but you get the idea. Phosphate group with these negative charges, very important later. It's what gives DNA their overall negative charge. Pentose sugar, anyway, these two together make up the backbone of DNA. You can see it's just repetitive units. Then you have the nitrogen bases hanging off the back. So there are two types of nucleotides, and it's based on their different nitrogenous base. So just like amino acids have the same structure, except for the 20 R groups, then nucleotides, there's only 4 kinds, not 20. And they're classified as either purines or pyrimidines. So you can cut pyrimidines out of something pure. That's something I learned from my genetics instructor. So if you notice, cytosine, thymine, and uracil, C-U-T spells cut, and pyrimidines kind of looks like pyramid. So you can cut pyramids out of something pure. You also notice that pyrimidines are a single ring structure. You can get 1 from a 2, but you can't get 2 from 1 without destroying the molecule. So something pure, purines, these are going to be your adenine and guanine. And then you're going to have pyrimidines, which are cytosine, thymine, and uracil, and they spell cut. So that's one way to remember. You can cut pyrimidines, pyramids. So when building a polymer or polynucleotide, again, you're going to have a long strand of sugar phosphate backbone, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then you're going to have this nitrogen base hanging along. So there's a polynucleotide right here. How does that bond? Well, you look at each one. Here's the individual nucleotide circled for you. And that red circled bond is what you call phosphodiester. That phosphate is bound covalently to 2 sugar groups. So it's a phosphodi, meaning 2. Ester is the sugar bond. So new base is added to the sugar of a previous base. And it just goes down instruction. Just like in amino acids, we had directionality going from the N terminus to the C terminus, and it can only be built in one direction, in that direction, N to C. Well, DNA also has directionality, and it goes from a 5' to a 3'. Okay, And so if you look at right up here in that corner, that was the oxygen, and you had the 1, 2, 3, 4, 5. That was the 5th carbon, and there's the phosphate group. So we call this the 5' end and come all the way down. And you see again, here would be the oxygen based on the previous picture. Carbon 1, 2, 3, 4, 5. But 3 is the end hanging out down here. So we call this a 3' end. So that's how we label DNA, 5' to 3'. All right, why is this important is a discussion for a different class, but it's important in short because this is the part that will interact within aqueous solution with another DNA molecule, polynucleotide, I should say. So as I said, there are two kinds of nucleic acids. There's RNA and there's DNA. Big differences here is that RNA is a ribose nucleic acid and it is a single nucleotide chain, whereas DNA is a double nucleotide chain. And as a double nucleotide chain, it has this helical structure. So the N bases, the nitrogen bases, bond in pairs across the chains, and that spirals the double helix. I referenced this discovery in the protein lecture, but James Watson and Francis Crick, who discovered the structure of DNA. DNA was known to exist, but we didn't know what structure it had. We knew it had nitrogen, it was a nucleic acid, but what nucleic acids look like, up to that point, nobody knew. Had a lot of evidence, and nobody could just put it together. And finally, in one serendipitous moment, these two, while off duty and off task, discovered and figured out the mathematics of the crystallography, and that it was indeed a sequence and it arranged in this double helix. I'll show you some pictures of them here in a few moments. So the pairing of these nucleotides is by way of hydrogen bonds. Again, a weak bond, but they're internal now. So if you have this one strand of DNA on this side, and you have this other strand of DNA on this side, you'll notice something. This side over here on the right looks like it's upside down. So they are what they call anti-parallel. They go in opposite directions, 3' to 5'. So once again, if you remember the oxygen, this would be our 5th carbon. This is 5', and down here's 3. Well, let's look over here, and we have oxygen 1, 2. This is 3 up here, and down here we have the 5' hanging. So 3 to 5 over here, 5 to 3 over here. That's what we mean by anti-parallel. They go in opposite directions, and it's beautiful. So it allows adenine and thymine to form 2 hydrogen bonds and to hold them in place. Guanine and cytosine, G and C, form 3 covalent hydrogen bonds, and collectively, although weak, remember properties of water. A lot of them are due just to their hydrogen bonding, the cohesion, adhesion, the high temperature stabilization, and so forth. All has to do with properties of those hydrogen bonds and polarity in water. Well, here we have another polar kind of molecule, and here's, you can see polarity because phosphate groups have negative charge. So DNA overall has a negative charge, but these NH groups allow these. So you just need one hydrogen of a polar compound, in this case a nucleotide, and any other atom, in this case nitrogen, of another nucleotide, and we have a stable structure in DNA. It's beautiful. If you need to listen to that again or go back and reread, you can. I won't test you heavily on it, but you do want to know there's these base pairing rules. T bonds with A, G's bond with C, right? And I think we know that much. It's very helpful to know that A and T have a double hydrogen bond and GC's form 3 hydrogen bonds. And why is it important? That stabilizes the molecule and keeps it oriented such that the inside of the DNA information is kept protected from the water and other elements. So it's an information polymer. It's a series of bases that encodes information like the letters of a book. Stored information is passed from parents to offspring simply by unzipping. Now that's what they were really after. When Watson and Crick discovered the structure of DNA, the big question was not what's the structure of DNA, the big question was how is it passed on from parents to offspring? What happens in that beautiful moment such that 9 months later this thing is crawling out and all over the floor? And how is it that it has each of your information? Well, they realized when they discovered the structure of DNA that that it was a sequence. And now it was a matter of finding out how it's replicated. So here we have in the cell, here's the nucleus with your 23 pairs of chromosomes. If we blow up one of those chromosomes, they're very large, long, billions of base pairs long molecules, that segments of which have genes on them. And a gene is what codes for an individual protein or a trait. So again, if we were to spread it out, you'd see that really the DNA strand is just really tightly coiled up. So double helix hydrogen bonds join between two strands of DNA. Look at that structure just for a moment. 1 nanometer halfway across in radius, 3.4 nanometers for 10 of them. So divided by 10 should be 0.34, and sure enough it is. And what that tells us, it's equidistant all the way up. It's not random, there's no bulging parts. You can follow that line straight down. It's a really pretty molecule. So how is it copied? Well, replication, you have two strands of DNA, helix, and they're complementary. So if you have one, you can build the other due to the base pairing rules. So you can see that if we had a GATGCA, the only thing the other one could be is a CTA, so on and so forth, right? Well, it doesn't take too much to realize that if we could somehow split these two strands and read each side of it, then you'd have two molecules where there used to be one. And sure enough, that's what happens. Now, it wasn't the first logical thought they had for how it replicates. There's three ideas for how it replicates, but this is the one that their scientific experiments settled on. So this is just kind of them, like any good scientist, they know to say, you always want more research, right? So all they really knew was the structure, but they could see that there was a sequence in that structure. So this is what they wrote in the original paper they published. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And that's what they want to know. So you can study that diagram. This is called semi-conservative replication. Semi, S-E-M-I, conservative. You conserve one strand in each of the two new strands, right? So this was synthesized from the environment. The foods you eat, the strawberries, the pizza, they used to be alive. Foods that were alive are made from living things. Chew it up, swallow. You have nucleases, enzymes that break it down, release these nucleic acids. So you don't get energy, you don't get mass, but sometimes you do need to replenish some of the nucleotides to make new DNA, especially if you're young, adolescent, or toddler, and you're in a very fast-growing time of your life. There's a lot of replication going on, so you need a lot of nucleotides to construct new DNA. He was 32, and he was 24. It's amazing. I mean, we see him as old people now, right? But they were young when they discovered this, and ambitious. So he has recently passed on, somewhat recently, about 8 to 10 years ago now. But he became really interested in consciousness and the dream states and things which are not front edge science. And so he never, I mean, he did do a few more good things within the field, but he became interested in other things of the brain. And he's still a good spokesman for science in his Cold Spring Harbor lab, and now he has become very interested in brain science and aging. There's the guy who helped him. He leaked the information to those two, and this is Maurice Wilkins. He helped develop the atomic bomb, and after that went off and it was announced they won the war, he felt so bad and guilty that he used his knowledge in science for death and said, I'm no longer going to focus on nuclear physics, I'm going into the science of life. And he actually did help to discover this structure. Well, he and his partner, the dark horse of the race, this was his partner, Rosalind Franklin. We don't have a modern picture of her, as you know, because she died just a couple years before the Nobel Prize was given. And she is the one who took the picture upon which he did the mathematics. And this is Crick. he's the one who put the puzzle together, that's Watson, and confirmed it. So together, these three received a Nobel Prize. Rosalind Franklin did not. Bless her heart, died of cancer. Nobody knew at that time what these X-rays can really do to you if you work with them day in, day out. Definitely should have and would have got the Nobel Prize had she been alive. Interesting note. This is for biotechnology. Take time to read this. This really has biotech implications. Maybe we'll reference it during the lab when we're doing our electrophoresis. All right, so another interesting note, something we're going to cover in this class is this structure. Let me move myself out of the way over here. Sorry. I want y'all to see this. So you might have heard that ATP is the energy currency of the cell. Maybe you haven't, but if you haven't, you have now. Adenosine triphosphate. So you have 3 phosphate groups. You'll notice this looks just like a nucleotide because it is. It's a pentose sugar, nitrogen base, and 3 phosphate groups. Well, these negative charges here repel each other, really pushing away. So this is a quasi, very unstable bond. But these covalent bonds are really strong. So it's much like a tug of war where you're being pushed in this case or pulled in opposite directions, but that rope is just holding you there. If someone came between you and the other team on the tug of war and clipped that rope, you get a lot of energy, right? And that's what happens. So this is a modified nucleotide, and these are very high energy bonds because of that repelling action of the oxygen, but then you have the covalent action between the phosphates themselves. So it's a very energetic molecule, and just by phosphorylating or dropping that phosphate group off with other atoms, you are destabilizing them. And destabilization means reaction. Reaction is energy. So very useful molecule for the transferring of energy. So that was about 17. I'm 2 minutes over. I wanted to review all the macromolecules with you shortly. You want to know and understand that carbohydrates are the energy molecule chosen by the body. They may not have the most energy, but they are the simplest energy, and they have enough that your body wants to use it. I liken carbohydrates to us. So, you know, we get hungry and we need something in us. We got to feed the beast. And so when we get home, we tend to go to the cabinets. What's in there? All Oreos, chips. We open the refrigerator, right? And pizza. We eat the leftovers. And once we've chipped it down and cookied it down and gotten all the junk food, and the hunger pains are gone. Well, then we can get out the steak and the potatoes and start defrosting it and cooking a real meal with real energy. And that would be like your lipids. So your body will always go for these simple sugars first before it goes to the complex ones, because simple sugars are, quite frankly, less easier and faster to break down. So the basic monomeric structure of carbohydrates is the monosaccharide, meaning one sugar. There are also raw materials for storage and things in the human body. We store it as glycogen in our liver. We might talk about glyconeogenesis later on, but your liver will store excess monosaccharides in the form of polysaccharides called glycogen. So we call this a glycosidic bond. And you'll notice again, it's a dehydration synthesis. So glucose, starch in plants, we can eat it, but we don't make it. Cellulose in plants, and glycogen in humans. So animals make glycogen, plants make starch. And it's all based on the same molecule, glucose, which is why we call it a monomer. It's the basic building block. Lipids, much more energetic because of all these covalent bonds. A lot more energy in here than in glucose, but it's going to take a lot longer to harness that energy. So this gray spot here is the glycerol, and you'll see it's 3 carbons. We got this oxygen, and this is then the fatty acid chain. So this is 1 fatty acid chain, 2, 3, so we can call this a triglyceride. That's your kind of typical lipid. Cholesterols look quite a bit different, but one thing they all have in common are these hydrocarbon chains, HC. So you do get a lot of energy, but because it's not used readily, we call it energy storage. We have to balance that diet, and when you're not totally carb loading all the time, you take it easy, your body will go to these lipids and break them down for energy. So it's, they convert the sugars, they convert the lipids into sugars that can be used. But if you're always supplying yourself with enough sugar and simple sugars, then your body never has to use these lipids, and that's when they become stored and we begin to gain weight, of course. But they're also used in membranes, and just like you learned of insulin is a protein hormone, there are other hormones that are steroid-based hormones like testosterone, aldosterone. So examples of lipids will be fat phospholipids in the cell membrane, steroids in the case of hormones. Proteins we just got through talking about, but you want to know their monomeric units or amino acids. Levels of structure, we had primary, secondary, tertiary, and quaternary, right? So 4 levels of structure, a variety of functions. The most functitious molecule, digestive enzymes, membrane channels, insulin proteins. They're pretty much the structural component and the functional component of cells besides membranes. And then this is the component of this lecture. But you can look at that slide and review for yourself. All right, well, I hope that helps. We are going to talk next about nucleic acids and how they determine proteins and just what the process is for protein synthesis.